So hi, just waiting for the presentation to open because I don't know why it's not. Share yet. I don't remember. Why? What? The file to download? Yeah, this we will use tomorrow morning, not in this session. No. No, not. No, today we are, I don't know what's going on. Today we will introduce all the tools that we will use together tomorrow, but in the next one, Felipe will start opening the program. So the ones that already have the program downloaded, it will be good to open with him and then start getting used. Yeah. Okay. Yeah, and the echo is much better right now, isn't it? It's good. It's good to understand with the mic right now. Okay. Yeah, we asked before if it was better with or without the microphone, but yeah, because the people from the back, they are they need the microphone. Because of that, we return to use. Ah, okay. For this, it's where it's bad for you. So, okay, this will be very fast presentation. I will just clarify some things for you to use with Felipe in the hands-on next session. So let's go this side. Let's understand a little bit more the names that we were talking about. The concepts, the main concepts you must understand to use the tools, and after to search for data and everything else. It's not going. Oh, let's stay here. So publication is something that everybody understands. What's the publication? Yeah, it's just missing. The definition is, yeah, there we go. The definition of publication is something that everybody, all scientists, all researcher has very clear in the mind. But just to be clear here, as we were discussing what we cannot process. We can process everything that has been published. And again, this is very important because we have some restriction of curators and some researchers that say, oh, no, I don't want my data to be online because this is my data. Somebody can steal my data. But we just deal with publication when we are talking about repositories and data extraction. So publication means articles or books. Dissertations are publications. Yeah, they are. Oh, yeah, but they are not published by a publisher. Yeah, but they are in the university. They are in the libraries of the university. They are publications. Reports for environmental consultings, they are publications also. Yeah, even extended abstracts, they also are publications. So it's very important for you when we are talking about data extraction, remember that we are just talking about data that's being published, but by being published, we mean all kinds of publications. Yeah, so this is what I'm talking about. The scientific publication then represents the permanent record of the collective achievement of a scientific community over time. So again, over time, when we are talking about legacy publication and the publication that we are also doing right now. Articles, books, theses, dissertations, abstracts, notes of communications. Peer review publications, also sometimes we extract that data from peer review publications. That's not so good because sometimes we have again the paper there when it's published, but yeah, okay. Taxonomic treatment, so this is the most important thing. Do you understand right now a little bit better after all this time we were talking about taxonomic treatment? Be true with me. Yeah, a little bit. No, no. Yeah, be true with me. Felipe said, and I said, but let's clarify taxonomic treatment. This is the key, is the important thing. What do we extract? This is always the question, and we say taxonomic treatments. So you must know, you must identify what's a taxonomic treatment. So Felipe used this definition, and I will repeat with you, and then we will see it. So to be clear, a well-defined part of a scientific publication that we just saw, documenting the features of a particular taxon. It can be a diagnosis of a taxon. It can include the reference to previous work, standard description, notes of distribution and habitat, citation of material, and general comments. So to be a taxonomic treatment, you must two things. You must a name of an organism and some information about it. So as I said in the morning, Julia Giora, and then from Porto Alegre is a treatment. I'm the organism. There is my name in the heading. And then there is some information about me. So this is a treatment. The treatment can be full of information or it can be just one information, but I need the name and an information. Now let's see if you really knows what I'm talking about. Oh, yeah, let's go here first. Example here. This is my paper, so don't, I'm just going here. This is my paper, so don't say it's bad because it's mine, okay? So here we have the name, Brachypopomus draco, new species. The name is here. And then we have information. We have the materials, holotype, paratype, non-type material, diagnosis, color, dimorphism, electric organ discharge, etiology, distribution, habitat. Julia, is all this one treatment? Yeah, it is, because we had one name, one fish species, and then all information. This is all one treatment. Very big, very complete. But now, let's test you. Let's start. It's too small? Can you read this? Can you read this? No? So let's make it bigger. Again, another paper written by me. Be kind with me. Yeah. How many treatments do you see here? Quiz. raise the hand and then pick a number. One? Everybody see one treatment? Let's see. Where it starts here. And then, okay, yeah. The entire name, Gymnotus refugio, is the species. And then, yeah, so this is one treatment. Yeah? Are we cool? Another one. Let's see. Bigger and bigger. Let's see. And here I have the paper. You see, the paper starts here. Abstract. And then let's go. Material and methods. No treatment here? No? Let's go just one page. Okay, where then? No treatment here? No? No? No, no? And here, no treatment? How many treatments here? Fingers. We have taxonomic names here, yeah? We have the order, family, subfamily, genus, another genus, and then where is the treatment starting here? Where? Tricky, no? Where the treatment starts? So do you think that this is a treatment? No. And this, no treatment here? No. Where the treatment starts? Where is the holotype? Here? Where? Yeah. Yeah. Of course. So this, you see how this is not that easy as the first one. Here we have order, but do we have information concerning order? No. And family? No. Subfamily? No. Then we have the genus. These are synonyms. There are the lists of synonyms, but then the synonyms refers to the genus. So here we have the nomenclature of my treatment and the treatment. You see? Mm-hmm. Yeah, if we have a template that is proper for this journal, for this format of the journal, the program will identify this as the heading of the treatment. So we have a heading. And then that, because we have a lot of taxonomic names here, you see order is also a taxonomic name. But there is no information about order. It's just a higher taxon of the genus. And the genus is what we are talking about. So the program will, and Felipe will show you after, will identify this as the heading of the treatment, the nomenclature. And this will be the reference group, will be the section where we have references to other studies. They are studying, they are quoting other studies.
No, yeah, you can hear me all right? Okay. No, yeah, I was just going to say that regarding your question is that the software itself has an inbuilt general template, sort of like when you run the macros within the software, you later see that it will try to guess where the most likely headings are. So, because there's a structure here, so genus, the name, the authors, and then it stops at the year. So it will generally predict that this is the start of a treatment, because that's different from having something like that with the page number and part him or other like information like description, illustration. It will often realize that this is not the start of a treatment, that this is an actual treatment. Sometimes it gets confused because some information is harder to fetch. So it might think this is a treatment because it doesn't interpret properly that this is a taxonomic name. It can happen. So it changes from publication to publication, from journal to journal. We'll get into more details tomorrow and later today when I do a quick step-by-step guide on how to operate with GGI, but this sort of answers your question and expands on it a little bit more. Okay.
Yeah, you see here the treatment continues. We have the description of that genus, everything referring for the same genus. So this is a long treatment, it's one. Another one, let's move. So this, let's go for one page. Okay. So again, we have new records here, material and methods, and then it starts here. And now let's do like that. Then you can see a little bit more. No. You see how many treatments here in this part, the second column. Fingers. Everybody from there. Everybody's with the scary face right there in that line. Let's try. So this is a treatment. Aranid is a treatment. Yes or no? No. Araniella is a treatment. No. But then we have Araniella villani. This is a treatment. Perfect. And here again, we have the higher taxon and then the treatment. Yeah? Are we getting a little bit more clear? And you see, this is very small treatment. We have the name, the nomenclature, the name of the taxon, and we have one reference group and material distribution, very fast, very small, but it is a treatment, same as the long ones. This is what we are looking for in the publications, please. Another treatment. Another treatment in the same publication. Yeah. Yeah, we have two. We have more than two, actually, I think. You see, we will have more and more in the same paper. We can have, in monographies, we can have like hundreds of treatments sometimes. Sometimes we have this kind of monography where we have the name of the species and distribution, the name and distribution, the name. It's a list. In just one page, we can have 20. But okay, it's a bit clearer, the concept of treatment. Yeah. Let's see if we have another one here. So this is something that we must understand because then it's too small. Too small again. Okay, so You know, you are very good because I've been teaching this for a while right now, and everybody tend to see the higher taxon and say, this is a treatment, this is a treatment, this is a treatment. Everybody does that. Yeah, so nice. And here, in this page, do we have treatments? One? Where is it? Buffonide? And there is no other treatment? What? Please not see. Oh. Here, are there anura? Is there a treatment? Let's see, it's bigger, bigger, bigger. This is a treatment, yes or no? Who says no? Who says yes? No? Why not? Because it's an order. And you, yes or no? No, why? Yeah, who else? Yes or no? Everybody thinks it's yes, why? Yeah, and it's an order. It's a problem for you? It's not a problem for you. It's not a problem for me also. Yeah, it could be. It's a taxonomic name. It's the order, but there is no problem. It's a taxonomic name. When we have higher taxon without information, so no problem, then there's no treatment. Because, okay, but I'm saying that it's Anura, Buffonide, and then the species. Everything is about Anura. No, it's about the species. I'm just saying the context where, but here I have a description for Anura. It's the order, and we say something about Anura. Anura include 17 bones. So I have information. This will go to the repositories, and when I go there and put Anura, I will have information from different publications, and different publications with, okay, this publication says just this about Anura. This is the definition of Anura. As I said, I am dealing with Systema Naturae, the Linnean publications right now. So we have descriptions for Kingdom Animalia. I have, yeah, we can, it's all, everything is okay if it is a taxonomic name and we have description. So this is good. One more, I think, one or two. Now this is not the last one. Let's see. So here, you see a treatment here. Different formats, but the concept is the same. Think about it. Do you see? Let's see. Here, no. And then here. That was just the name, the title, but since this is a chapter inside a book, sometimes we have something like that. We have the title, and the title looks like the name of the treatment, but no, it's just the title of the chapter. And then inside that, we have this. These are treatments. Yeah, we have a treatment here that is for this species. This is the nomenclature, and we just have treatment citations, previous treatment citations, just this, and this is enough. And here we have another one. And just two. One is this species, Carineta fasciculata, and there is just one section that is this. This is the reference to previous treatments, and this is the other. So I have here one treatment, and I have here another one. Two treatments. Clear? What's your problem with those? No, they are just different formats. You see, so this is like you think it's easy, but even us sometimes we have doubts and we work in home offices, but we work all together. We have a chat. Sometimes the guys say, oh, people, I have this. Do you think this is a treatment? Because the formats are so different and there is everything happening in publications, in different publications. You can have doubts. So it's good for us to have here some different things. And then now, when you are looking to the papers you are reading for your research, try to pay attention and make these exercises yourself. Is this a treatment? Where the treatment starts? Where the treatment ends? So let's move a little bit. Worry me. So another thing that we are always talking about when we are talking about the extraction is the specimen. So the definition of a specimen that's different than species is a gathering or part of a gathering of a single species or an infraspecific taxon, mounted either as a single preparation or as more than one preparation within the parts clearly labeled between. So we are talking about collections right now. Everybody works with collections. So I am a zoologist. I work with vials with animals. And if you are botanical, you will work with the plates with the plants. So if I'm talking about the animals, I have one vial. This is a specimen. Can have more than one fish inside, no problem. This is a specimen. And as a botanical, you know that you don't have the entire plant sometimes, but you know, specimen. Think about the collection, and then you'll be cool with the concept. Yeah, you see? This is my fish. Another thing that we are always talking about, material citation. Material citation, if you go very literal, you never go wrong. Is when you are quoting, we are citing the material that you are using to make your description. So a reference to or a citation of one part of multiple specimen in a scholarly publication. So when we were looking for treatments in the publications, we had the material examined. These are the material citation. Then can be one holotype, just one paratype, and then you have one collection code, another collection code, and you have more than one material citation. So the difficult thing when we are extracting data, and that you see with Felipe, is to define where a material citation starts and when it ends. To know that this is a material citation is easy. Okay, the difficult part was to identify the treatment, but the material citation is easy. The difficult part is to define the limits, the boundaries. You see, some are very obvious, some are not so obvious, the amount of information, but you see where to stop. Here we have holotype. Of course, we have just one material citation. It's one holotype. Everybody knows that. But here we have paratypes. Look at this. Not so easy. But we have to split it in the Golden Gate. So we have information to different material citations. Here we have in the paratypes. You see the same one I was looking. You see? So if we don't have a proper separation, as do you remember in the first lecture in the morning, I was talking about how to publish in the future to be machine readable. One very important thing is to be very clear with the separation of the material citation. So right now, Zootaxa does, and Zootaxa? European Journal, Zootaxa does also, no? European Journal of Taxonomy does. They use the bullet points. What's the other name for bullet points? Bullet points good for you? The small circles? Yeah, between one and the other. So yeah, this is very machine readable. GGI would never get wrong with that. But without this kind of separation, we have to look for patterns. As I said, I am a mother of two. So when they are small, oh, well, let's play like, choose the pattern. Yeah, you see, yeah, red and yellow, red and yellow, red and yellow. Yeah, red and yellow and green. This is a pattern. I have to look for patterns. So yeah, in this, when we have a collection code, I stop. I have another material citation. Or here, when I have a location, yeah, Brazil, then Colombia, Peru, Ecuador, I stop. I have another material. Or a specimen count, 4 specimens.
So this is the bullet point that...
Yeah, the bullet point.
Yeah.
Here we have the bullet point, much better. But there is another way. And this is very important because after we will parse the material citation. You will do that in the program. So we will open each one of the material citation and we will have all the definition, holotype, and then number, specimen count, country, location, collection date, collector, what's the term? Collection method, collection code. I will have all this, and then I will have all the information, the metadata associated to the material citation that goes to the repository. You see all the beautiful things that we were talking about, all the reuse, all the information available must go through all this process to be identified by the program and to go. So if we don't have a specific boundary definition, it's very difficult for the program. And then after that, with all this parsing, we can go to the repositories and query, as Felipe showed a little bit, and we will see on Thursday, how to query. So you can just go for collecting year. How can I have this information? Because I had the definition of the treatment, the definition of material citation, then the boundary of the material citation, the parsing of the material citation, and then I can query and have this information on my hand. So it's a process, and you must understand the steps. Because of that, I said that we were climbing stairs, and we are going there. One more definition for the GGI subsection. So I said in the morning that we had a paper and we would split the paper in many parts. So that part we call subsection. Oh, Julia, why not section? Because here we have more botanists than zoologists. Then you know that section is, it's a classification for botany. So it would be like tricky. So we don't have sections. In the paper, we have subsections. Subsection is everything that's outside the treatment. We have one subsection that is title, another that is abstract, another is introduction, another is material citation. Why this is important? If we just export the treatment, because do you remember that we have other repositories, other that use the query, like Felipe, help me with English, like biodiversity PNC, we have the query, continua, uma pergunta aqui para o meu Google. Okay, full text search, this it's not an English problem. It's like an age problem. The words just go away. Yeah. Full text search. So since you have full text search, the search goes inside the paper and you must have the subsections defined. But then, oh no, yeah, subsection. Yeah, there is a subsection. If we have subsection, it's outside the treatment, but we have the treatment. So inside the treatment, it is subsection. So we were looking for treatments and we saw some with a lot of subsections. We had material examined, we had etymology, diagnosis, descriptions. Each one of these are subsections. So to be a treatment, there is one condition to be a treatment. Now that you know these names, how many subsections you need at least to be a treatment? It's a good question. Only one, do you think? But one subsection? Guesses, please. Louder, speak louder. The guys are discussing, but they are just between each other.
You've already answered this before, but with a different name.
Yeah.
It's
You answer, but not with the name. No? We need at least two subsections because we need one that is the nomenclature. We always need the nomenclature, the name, and then at least one more. Because we need a name and something about that name. Something can be just one, but can be. Or everything. If I had just nomenclature and materials exam, okay, two subsection. Treatment citation. Now, what's a treatment citation? It's a link to a citation of a given taxonomic name which is related to in the same rank of the treatment there it's been referred. Yeah, it's complicated. No, it's not. We've seen it in the treatments. It's when we have the treatment, we have the nomenclature, the name, and then under it, we have other names that were attributed to that and with the reference to the bibliography. So we are saying, yeah, we have been studied this taxon before here. And this is very good and very important because we will have repositories just to query this, the treatment citations. So here is a treatment a little bit like it can be in GGI with the nomenclature, then all the subsections, now treatment citation here, you see? Treatment citation. You always have a citation to another paper. Yeah, and also as we parse the treatment citation, we have metadata associated to it, and then it's very important because we can query, we can see, oh, this taxonomic name has been quoted in other treatments, and then we can have a synonymic list and all that based on the treatment citations we have. Oh, then this is very easy for you, species distribution, the range, geographic range, and the record, just to be different between the other. So the species distribution is the geographic range. It will be in the distribution, part, subsection of my paper. But then we have just the records. And this is something that we will look for in GBIF, for example. If we look for the records in GBIF, we are just looking for that records that came from our material citation, from our treatments. So this is introduction for you then for you to be able to start working with Felipe in GGI. Is this clear for you? Treatments, material citations, subsection, this must be Yeah. So thank you very much. Go there.
So again, everyone, will be the last one for the day, so everyone can rest after that. I know it might be a bit too much. It's a lot of content, and usually to take that into one day or even 1 week, it can be overwhelming. We understand that, but it is the nature of the workshops that we have limited time to do so. But again, as we've been saying, to whoever wants to keep collaborating and wants to engage in that, you have time and space to get to properly know the tools and uh, dive deeper into what can be done. Uh, Please don't feel discouraged by the amount of content, okay? What I'm going to do right now is just to quickly go through the first steps into getting to use Golden Gate Imagine. So you can, as I mentioned before, you can just unzip it and start using it. Uh, I think that most of you have downloaded it. I know some of you will have Macs. The guidance here is for Windows, but it works on Mac. Is just slightly different. I might have to talk to you in person uh, just to help you out, but it tends to work the same. It's just that the first steps might be a little bit different in terms of actually opening it. I don't remember right now what's the file that needs to be done, but anyway, to use it, you download it. It's a zip file. You unzip it to whatever directory you want to have it unzipped. We'll unzip everything from in that folder, and from then on, you can open the executable file there. Uh, I I think for Mac is this one actually is, if I'm not mistaken. It's the jar file, but I have to test it with someone just in case. Once you open the executable file, it will show you that screen there, that small window. Please click yes. This is a node thing for DJI because when the internet was not as fast when we first started this is like a legacy thing that some people had trouble connecting it. They wanted to use DJI without connection to the internet, but that's just for a startup. You don't need to keep the internet connection later. And then you're faced with this window. If you don't have internet when using it, and you can do that, you only have the first two options. Uh, Always use either default Imagine or in both iterations. If you have internet, go for the third option because it will update to the latest version if you do so. You can either double click on it or click once and then OK. Once it opens and updates, you get this screen. This is DJI without any PDF being loaded to it. So to open a document, you from a local device, you click on File, Open Document, and then you can choose between opening a born digital PDF or scanned, or even an IMF. What is an IMF, you ask me? That's the image markup file. Whenever you open a PDF within Golden Gate Imagine, that file is decoded into an IMF. An IMF is sort of a zip file, so in the actual format of it, it concatenates several files within it, but that is the machine actionable format of the files. And once you have that, you can save it as an IMF on your local drive to work later. Sorry. To continue working on different sessions. But that's also the format that it gets exported to the server once you do so. There are many options in there. There's a help button if you want to have a look. That's usually used for scanned documents more often because there are different types of scanned documents. Some of them have OCR built in them, some don't, so you need to select accordingly. You can see the options here, so or digital scanned text layer digitized. It depends on the file that you have. Okay, this currently is not that important, but you can open directly from the server if it's for the it's not important for the purposes of the hands-on, the start of the hands-on experience we'll have tomorrow, but you will be able to load a file directly from the server so that you can edit and improve it. Okay, for that you need a login. That's not a hard thing to do. You just need to ask us for a login. Then we can give you one. Trouble is I can't do it myself. I need to ask our programmer to do so. So please, if you want a login, please do ask me today if you can, and then tomorrow we can have it ready.
Felipe, let me just. Yeah, this is part of what we were talking with you about the after the course. As I said, we have the continuous training, and we can show to you how to enter the Plazi page that was not working today, but tomorrow. And then we enter there with you. We have modules in the Google Classroom, and we have another platform for the ones that's not okay with Google, that is also a learning path. And then you can follow all the classes for individual extraction, for quality control. And then after that, we schedule a meeting. And then we give to you the user and the password to use the platform and the program as new users. So we can chat a little bit, we can have some doubts solved, and you start using the program. Then we make extraction. When we save the extraction, we will receive, Felipe can go there every day and then see the new users upload. We go there and we see if it's everything okay. And if it's okay, we send it to the repositories. All the extractions that new users the new user do, they never go to the repositories without our approval. So if there is something that's not okay, we return to you with a feedback. And we have like 7 levels. Like, oh, this person is okay extracting metadata. This person is okay parsing material citation. Since when you are getting very good in one of these things, we liberate the certificate for you. So you are certified to liberate metadata. You are certified. And in a while, we will be fully certified. Then you can do your extractions and it goes directly to the repository. So it's very important to say at that point that right now you don't have a user and password. We can open with you, but just locally a document. But if you do the extraction, it doesn't go anywhere because you don't have. But if you want to do that, it's very possible. It's just a matter of talking with us and then continues your training. And this is it.
Thank you, Judy. So I'm not sure if we should go too much into the detail, but this is just to showcase the amount of annotations that are featured in GGI. There's more than that. This is just a few of them. It's like there's a lot of layers that can be found. Like you can organize paragraphs, you can organize blocks, lines, etc. Like images, tables. You can add captions. So we've been talking mostly about treatments, but there's more to that. Remember, we were talking about images and tables before, so there's the whole, like the parts of it are also annotated within DJI. So what happens sometimes, for instance, with tables, I'm not sure if we have any here as an example, a lot of publications, especially in biodiversity reports, have their data completely in tables. So well, before, like until 2024, actually, we didn't have the means to extract data properly from tables. Now we have. So you can have treatments completely within tables. We structure them in a way that it's much easier to extract the data from. It was one of the hardest places to extract manually. If you so wish to, if you go to a PDF right now, try to open and get the data from a table. It's really annoying to copy over. Like it's one of the hardest things to do. So that's one of the things we've managed to improve. So those are other annotations that can be that will be featured within the software. Okay. Um, so let me come back here. So where's the okay? So we need to one thing we need to do with DJI is always make the document structure. That's one of the first tools that we use, and then we add the metadata. We see the images and graphics. They all have attributes. They have their identifiers once everything is gone through the process. Tables here. We can fix the word flow. Again, I'm just going quickly through this because the actual hands-on is tomorrow. Is just for you to get a glimpse of what is to come. Okay. So we can manually edit the workflow of the documents, the subsections and sub-subsections as well. We can all edit it that because sometimes what you might find is, for instance, a treatment that's completely in a paragraph because sometimes authors tend to concatenate the information in one single paragraph, but there's information there, but how do you make it so that the treatment that is within one paragraph is turned into an actual treatment, a proper like reusable treatment that needs two. So you have to manually break it down. So you tell the software manually that usually can't be done automatically. There are ways, but you need very precise and constant uses of separators by authors, which tends not to happen, to tell like, oh, this first part of a line is where the nomenclature ends. Now it's a description. Now it's a material citation. So usually having everything in one paragraph is not good practice. But that being said, we are faced with that and we just have to deal with that. But that's one of the less automatable things to do. Okay. Okay. Don't need to go through this. Not this as well. Okay. The annotation boundaries, that's one thing that Julia talked about before. Everything can be manually fixed. There are many tools that we can use to fix that. And there's even tools where you can remove and add several annotations at the same time. So there's one tool called Annotate Similar. You can look for patterns and use regular expressions to find, for instance, accession numbers. I want to annotate all accession numbers within one document. I find a pattern, which is usually 2 letters and a sequence of numbers. So you can do that. Okay. And then at the end, you save it. You either save it to locally or save it to the server. And then you end up with the persistent identifier if you save it to the server. I know this was very quick and very not clear what's going to happen tomorrow, but it's just so that you get ready to have the hands-on. Any questions right now? Who will want to be will take part on the actual hands-on? If you can raise your hand, please. Okay, good. So I think that tomorrow we will have 2 groups probably. Julia and I can do half, where each one of us helps you, half of you, and the other one does the rest.
Or we can do like that. You do everything
Yeah,
and
no,
then looking there, and then I will be walking through
yeah,
the
that's one
classes
option, yeah.
and helping.
Because
So
I'll have a demo, okay? I'll do an
yeah,
actual live demo here
yeah.
on the screen. But whoever, all of you that raise your hand, please, if you don't manage to get it running today, please come to me and we'll talk right now. And then we can get it set up for tomorrow, okay?
Yeah, what we do is like this. We open a paper and we all do the same. And then
The
Felipe
same paper, yeah.
does one step. And then I said, now everybody does. Then I wait a little bit. Problem. And I go there. Go there.
Yeah,
Another
from
step.
previous experience, it's not a good idea to everyone to do their own because it creates a lot of problems. For first experience, it's better if all of us do the same thing, okay?
Yeah.
But of course, during the week we can do more. It's
Yeah, we
just a
need
matter.
much more time if everybody will do one, and then less people and more of us.
Yeah. And some people have do things faster than others. It's just natural, but okay. No, let me go have a look. So, 'Choose and detect document. Oh, sure where's everything else? No, that's. I'll need to go through it with you. Maybe you maybe use local master configuration. You need to use the Fold Imagine to run it. So maybe restart the software and use the Fold Imagine. So yeah, that's actually key. It's a good that you showed that to me. So guys, please remember, there's 4 options here. Please do use the default imagine options. Either the local one or the host one, doesn't really matter. But you do need these two, okay? For the purpose of the workshop. Yeah, it's default.imagine. Okay. Yeah, I think that's. Has anything else to ask for now? That we're all done for the day, but we have time to talk person to person right now. We can go. Yeah. So yeah, to do that, you need to tell me like just send me your name and or what you would like to see as your username, and then I can ask our programmer to register you. Okay. We actually, that's a good thing that you bring this up because we had an experience in South Africa that we decided to create the usernames ourselves, and some people weren't happy because of cultural reasons. It was a bit offensive for their names were a bit changed, so it was important for them. I'm not sure if this would happen here, but I prefer if you guys choose your usernames. So like, for instance, mine is just Felipe, my username. But of course, if there's more Felipes, then they would need to do Felipes and whatever, something else. Okay, but just write that down to me and I'll send it to my programmer to create the username for you, okay?
Yeah, but again, for tomorrow, we don't need passwords and
Yeah,
username.
tomorrow we are doing local.
Yeah, we are doing just local. This is
The
just,
local the local use you don't need password.
yeah.
Just to download from the server to up or to open an already processed article, okay?
We need the password just for accessing the server, Plaza server and treatment bank.
For
Just
upload or
access.
download or
Just to
reuse,
send, yeah, send the data for the
or
repository.
editing. Yeah. The latest. The latest version. Yeah. Go ahead. I think it's still off, which is a bit weird, but Yeah, yeah. Yeah, it's the one that I showed. Yeah, yeah. Yeah, it's one of the files in the root folder that you need to open. It's just one, yeah. Yeah, it's gg imagine dot exe. That's the for Windows users. Again, I know that I've seen some Macs around there, so that you need to use the .jar file. Yeah. Oh, yeah. Actually, let me have a look at yours.
As I said, we had the course in Google Scholar that you can access through Plazi website, but I don't know why the website is off, you see? But there is another way that is, this is a partnership with the Swiss Bioinformatics Institution. Also, you can Google Elixir, test, Plazi, uh, and then you enter there. I can send you the link through the organization also. And then here we have the learning path. We have introduction to Plazi workflow, fair concept. And then if we enter here, you have the presentations, the lessons, then how to process and curate it, what we are starting right now and we will be doing tomorrow. Then we have here, getting used to Golden Gate, individual extraction, all the material, a glossary for GGI, a lot of material, and it's open, available for everybody. And then the reusing, again, the same. In classroom, we have the same classes, and we also have a space with exercises, so you can contact us, do some exercises in the middle. And then this is the way we do to have the certification, to keep you getting used to DGI and to be a extractor or a data curator. Again, you don't need to extract data, but if you understand GGI and if you have the certification and you are using GB for your query and you see something that's not right, oh, the location is wrong, or my affiliation is not there, you can do both ways. You can contact Plazi and you can report this problem. I will show you how to do it in another day. And then we fix the problem. We do that all the time. Or as new users, you can enter GGI and fix by yourselves very easily. And this is very good when you are working with projects, with studies, students. Okay, I have a project with a student to fix all data from India concerning fish. Then you enter there, you see what's not good. You don't have to extract the entire paper. You just fix the little things and then you curate the data. And this is very useful. So this is the material that we are using here and plus a lot of material. It's in this learning path. I will send you the link. And when Plazi is working again, I will show to you the other way to have the material and then increase your knowledge about distraction and have the certification. I just wanted to show to you because some may go to the house and then look a little bit more to have more questions. Oh, I don't understand. Oh, Julia spoke something very fast. So you can go there and come tomorrow with very fresh questions.
Thank you very much. Well, again, now that we've wrapped up, I think you have something to say as well. And then, yeah, we'll talk after we go down. Okay.
And I guess with that, we come to the end of day one. And today we explored about India biodiversity landscape, the principle of fate data, and the tools that are making data liberation a reality. And we hope this session have sparked new ideas and connections for all of you. We look forward to your enthusiastic participation for the next 3 days as we go deeper into the hands-on workflow and collaborative exploration. On the behalf of the entire organizing team, thank you for your energy and engagement today. Have a wonderful evening and we will see you tomorrow morning.